Hello.
Yes, I'm back.
It is a great pleasure to introduce Professor Wolfram Schultz as our first invited speaker. Wolfram is, by the people, if you've heard of dopamine, you've heard of the name of Wolfram. So he is the person behind. You have to do something about this.
There's a
آقای
feedback.
پنجه‌پور. میکر- صدای داخل رو خاموش کن چون بچه صدای منو میشنون. اسپیکر رو خاموش کن. من صدام برایشون روشن. Wolfram, can you hear me?
Yep. Yes.
So it's a pleasure to host as a first speaker Professor Wolfram Schultz. Wolfram is one of the most well-known neuroscientists in the world. He has worked in the field, I think, for more than 20 to 30 years. Lots of publications, lots of citations. He's mostly known for his work about dopamine and the firing of dopamine neurons. And he is the person who has discovered the basically the role of dopamine in what we now know as reward prediction error. So all of the research that you hear about reinforcement learning, the actor critic model that is now widely used in both neuroscience and also in computational neuroscience, it's all stemming from his work. And so he is now currently a full professor in Cambridge University. And he has a long history of working with non-human primates, recording from dopaminergic regions. And he's also very interested in neuroeconomical interpretations of choice and dopamine firing. So today we have the pleasure of hosting him, and the title of his talk is "Scalar Reward Signals for Vectorial Multi-Component Choice of Options in Monkey Orbitofrontal Cortex." He's also recording from Cortex, as you can see, and basically we are all ears, Wolfram, to hear your talk and learn something new today. Thank you.
All right. Good morning, everybody. Yes, indeed, it took not 20 or 30 years, it took 40 years. I'm working on dopamine for 40 years, 45 years. And it took us 10 years to figure out the prediction error, another 5 or 10 years to get it properly published. And when we saw these dopamine responses, we couldn't believe it. And we thought, of course, we were making a mistake. And so we went to other brain structures, like the striatum and the frontal cortex and the supplementary motor area. And of course, we found completely different signatures of neurons. But since then, my interest stayed with frontal cortex and striatum. And when I discussed with you guys what to present, I said, well, we have done recently some quite extensive work on the monkey orbitofrontal cortex and on humans with a particular economic question that we find is important to understand also the dopamine neurons. And this is what I'm going to talk about. I understand you can hear me properly. Can you hear me?
Yes, I can hear you.
Okay.
Clearly.
All right. So here we go. The work I'm presenting is largely, but not alone, the work of Alex Pastor Bernier, and it was helped a lot by my friend Charlie Plott from Caltech, who is a very renowned economist and very interested in experiments. So let's go. Sorry. This is the dopamine signal that you may be familiar with, the reward prediction error. At the top, an unpredicted reward elicits a excitation in the neurons. A fully predicted reward elicits no response to the reward, but a response to the stimulus. And a reward that is absent, but has been predicted, elicits an inhibition in the dopamine neurons. There's a lot of heterogeneity in the signal, but in a nutshell, this is what you see in 95% of VTA dopamine neurons in about 60% of substantia nigra dopamine neurons. And as although this is very straightforward and easy to understand and completely understandable in a time-sensitive Pavlovian learning scenario, the later analysis or the later playing around showed that there is in fact more to the dopamine signal. And if you look at the response marked by CS2, if you have another stimulus that predicts a reward predicting stimulus that then predicts the reward, you get a response to that stimulus. And that is very typical for a category of neuronal reinforcement of reinforcement models called the temporal difference learning model from Sutton and Bartow that goes back all the way to the Bellman equation that deals with reward maximization. The TD model is in fact a solution to the temporal difference differential equation. Okay, so when we have this, before I go further, I should mention some developments that have become quite well known in the current times, and that is that there are multiple dopamine signals. In fact, what we are seeing is the reward prediction error signal as shown in red that can vary as shown by the dotted lines. It occurs both with reward and with any kind of stimuli. Okay. What in addition we have found, and that has created a lot of confusion, or it has created a lot of confusion with us, and we try to solve it. An initial response to these phasic responses, and that's a salient signal. So people have always confused or often confused this, and we can find this separately. I'm not going into the detail. All right. And then what also happens is that when you have big movements, not small movements, but big movements that generate behavioral activation and a lot of sensory stimuli, you get a much smaller dopamine activity that's also called a ramp. And of course, you have tonic dopamine level that you need to prevent Parkinson's disease. And I would also like to point out to the time course, we have a very short time course. The phasic dopamine signal that does a reward prediction error is always shorter than 1 second. And anything that's longer than 1 or 2 seconds has probably a mixture of these signals. So the idea is that one brain system, we're used to this about the visual cortex and the motor cortex, one brain system is one function, and that may well be true for some brain systems, but it's certainly not true for the dopamine function. So if somebody asks you, what is dopamine doing? You can only say, well, it does this and this and this. There's not one function, and when you find another function, it contradicts absolutely everything. Okay? Just as a update from the current situation. Okay, we're going to do this here very briefly, and we have this signal, and then we have two problems. The first one is that reward value is subjective. Normally people vary the magnitude of reward, but that's a physical change. But the reward value is subjective, and we need to have a subjective value updating signal in order to be efficient or effective. Otherwise, the signal needs to be translated afterwards into a subjective signal that becomes very complicated. And what we have done is we have addressed the question and found a subjective reward signal in dopamine neurons. I'll show that very quickly. And the second thing is that reward have multiple components. An apple has taste and size. And your reward value for the apple depends on both of these components or scalars. So you have a scalar signal, reward responses only go up and down, neuronal responses only go up and down, but the reward components are a vector, or the reward is a vector. It has one or two, it has 2, 3, 4, 5 scalars, and you need to have this vectorial to scalar transformation in order to get a scalar reward signal. Okay, and that is the main talk. So very quickly, the subjective value has been addressed in economics for hundreds of years, starting, for example, with Daniel Bernoulli. And we're going for this. This is particularly the utility, which is a mathematical function. You can get temporal discounting and see the value and the value response of dopamine neurons are subjective, but that's not a terribly satisfying thing. You want to have a very general idea of subjective value, ideally as a mathematical function, and utility presents this mathematical function. So we have estimated in a particular behavioral procedure, there are several procedures available, in this case, by the so-called certainty equivalent in risky choices, we have estimated a utility signal, and the utility signal initially goes up very rapidly, which indicates risk seeking, and then it gradually flattens, and then it becomes concave, which means risk avoiding. So if you have a reward prediction error signal in dopamine neurons, it should actually follow this curve if it's subjective. So what we do is we present 3 gambles to the animal, one or the other, one out of 3 gambles, and the 3 gambles produce the same physical reward prediction error. So I have to explain the top of this bar. The top bar indicates a large reward, the bottom indicates a slightly smaller reward. And here it's an even smaller reward and an even smaller reward. And these rewards occur with 50% each. That means if the animal is presented with the screen stimulus, it will receive 2 seconds later either the larger reward or the smaller reward. It will receive one, but only one of these rewards. So this is a gamble. This is uncertain, 50-50. And then we have 3 gambles. They have all the same difference between top and bottom, but they are positioned at different levels, at different so-called expected values of these probability distributions of the 2 rewards. So the physical reward prediction error is always the same. The RPE is always the same. And when we look at the positive one, it should be the same. If the neurons code the subjective reward prediction error, like the utility prediction error, then of course that would depend on the steepness of the function. Here the steepness is, the slope is very flat. Here the slope is steep, and here the slope is flat again. And that's precisely what we see. Here we take the dopamine signal, the reward prediction error, we look at the positive reward prediction error response, and when we see this, then we find out that at the steep part of the utility function, the prediction error response of the dopamine neurons at the time of the reward is much larger than it is at the other parts. So we see a utility prediction error. Dopamine neurons are coding utility, and we can rephrase Riskola-Wagner and the TD model in terms of utility. That's not important when you do machine learning. A computer silicon machine does not make that difference, but for biology is absolutely crucial. Okay, that's and then we can do a little test here, and this is a wonderful risky gamble presentation. Here, this is a much more risky gamble because you don't know whether you get a very large or very small reward, or this is a less risky gamble, right? Mean is the same, there's no skewness, but the variance is different between these 2 here. So this is a riskier one, that's a less risky one. Then we plot our utility function. That's from a different animal. It looks slightly different, but the characteristic is still the same: convex, linear, and then concave. And then we plot these 2 gambles here in the x-axis, and then we calculate the expected utility, because we go over on that utility function, and then go to the left, and we see that the riskier gamble, the red gamble, has a larger expected utility than the less risky gamble. And that means the animal should, if this mathematical function has any predictive value as a mathematical function, it should indicate that the animal should choose this risky gamble more frequently than the other, because it has a higher subjective value. And that's in fact the case. It's not 100 to 0. It's a stochastic choice situation. The animal chooses sometimes the dominated option, but overall the animal prefers the riskier gamble. That means the animal shows by its choices that not only the utility function has predictive value, but also that the red gamble has a higher value for the animal. So what do the dopamine neurons do? If they would code the physical value the same, then that's the same mean. The response should be the same. If they code the subjective value according to utility, they should have a stronger response to the red gamble, and that's in fact the case. This is not a risk signal, it's a value signal that reflects the larger utility of the more risky gamble. So dopamine neurons actually do, and we have plenty of other tests. The dopamine neurons go for utility, and it's utility that the animals are maximizing, not physical value, as this slide shows. All right. Okay, now we will go to the 2 reward components. That was our second problem. Rewards have different size, probability, taste, color. So rewards are vectors, but neuronal signals can only go up and down. All right. Neuronal signals are scalars. That's my vectorial to scalar transformation I mentioned a little earlier. So the neuronal reward signals that are worth a name, a real neuronal reward signal, should process the vectorial rewards. It's absolutely necessary. Otherwise, a reward signal wouldn't be worth it if you only codes. It wouldn't be worth anything for reinforcing decision variables and neurons that code decision variables if they only go for one component, like for the color of the apple. You cannot make a choice alone between apples alone on the basis of the color of the apple, right? So how can neuronal signals do this? And you approach this in two steps. Or we did approach it. First, we get a value from each apple, from behavior. We infer value from choices. And that value, of course, only goes up and down. So the value, the subjective value, the utility is a scalar. It's a single dimensional metric. And then, of course, once you have that single dimensional scalar metric for value, then you can find a scalar neural value signal. So I'll show you. Okay, here is my apple. The apple has a particular size. It's not terribly big, but it's very sweet, right? You may like the apple, maybe not. Maybe you like that apple. That apple is much bigger. You have more apple to eat, but it's not as sweet as that, okay? So then how do you compare these apples? Which one is better for you? Well, you can only equilibrate the two apples at choice indifference, and then you can see how much of the large apple is worth how much of the smaller apple, right? You can give up some amount of sweetness in order to get a larger apple, but you don't want to lose anything in value, right? The two should have the same value. We infer value, subjective value, from choices, and if two objects are chosen equally frequently, we say they have the same value, right? When you choose between these two apples and you say, I cannot make up my mind, 50% I take the red apple, 50% I take the green apple, well, they're probably have the same birth, right? Okay. So same value, all right? Although the two components vary, the apples have still the same scalar value. Now we can have an apple that's a little bit bigger than one apple and a little bit sweeter than the other apple, the yellow one. And then these signs here, these little triangles, show preference. That yellow apple is preferred to the red apple. It's also preferred to the green apple. So this yellow apple seems to have a larger value, all right? Okay, higher value. All right, so we can then do this vectorial to scalar transformation by defining the scalar utility, the scalar value as a function of the two components. As a function, that means any weighting would do the job, whatever it fits best. Okay, so all right, then let's go and do a proper experiment. We have to define what we're doing. We do discrete choice between two options. They're collectively exhaustive option set. So there are two options usually that we use, and all options are included in the options. There's nothing hidden. The two options are mutually exclusive. You can only get one or the other. They appear simultaneously, which avoids any adaptation intervening stimuli, which is an interesting situation to have separate presentation. But for our case, it makes things more complicated. And the options are distinct and separable, very clear options. And we keep the cost constant. The cost is a part of the subjective value, but we don't want to change that, okay? So, and now we infer the subjective value from observable choice. Subjective value you cannot measure. You can only measure choices. And from the choice, we infer the subjective value. And as I said, equally frequent choice indicates that value. And now we can go with these ideas to our two-component choice options. And again, we need to make it simple and symmetric. That means we use two juices as option, like taste and size of the apple. We use distinct rewards, like the grape juice and the blackcurrant juice. In each option, each option has exactly the same two rewards, but at different amounts. Okay. So here we go. We have a reference option, a variable option. We do psychophysics. We give the animal a choice between a large amount of blackcurrant juice and no grape juice, and a small amount of blackcurrant juice and some grape juice. Okay, this choice. And the animal will choose the variable option very little, because the blackcurrant is quite small. Okay. And then we increase, you can see this here, we increase the blackcurrant a little bit, and the animal should choose this variable option a bit more. Okay, this is here, and it does it. It chooses more. On the right-hand side, we'll see the choice probability. Okay, all right. Now we move this again here. We go a little higher with the blackcurrant, and we continue. And at one point, we have 50-50 choice probability approximately. So they seem to have the same value now. And we continue, we go higher and higher and higher, and then we fit a psychophysical function. Very straightforward technology, okay? And then we can plot this. We plot our blackcurrant juice, we plot our grape juice. We see that they are equally frequently chosen at some amount of blackcurrant juice and some amount of grape juice. And then we say they have the same value, as I mentioned. And the animal is, in this case, giving up some blackcurrant juice in order to get some grape juice when he goes to choose the option with more grape juice. Okay, and then we can do multiple of these points in between, and we fit a curve, and it turns out they're mostly convex and maybe a bit linear. And then when we have higher reward amounts, they produce a higher value. But then again, we can get an indifference curve in which all amounts, all options have the same value. All right. Okay. So this is the scheme of these indifference curves. Same value along curves and increasing value across curves. All right. Now we have a metric in which we can look at neurons. Let's see. We can use these indifference curves for different juice combinations, blackcurrant juice versus grape juice. Then we can enhance the juices with some taste enhancers, monosodium glutamate, and so forth. We can use water. The slope is slower, but it's all the same. Then there is this situation with the yogurt, and that's an interesting situation. We have to give more blackcurrant juice in order to have choice indifference against yogurt. This is in a monkey, right? So remember, yogurt, these curves. If you, like I, hate plain gray yogurt, I need a bit more in the yogurt in order to eat it. That means I need, in this curve, some additional, in this case, blackcurrant juice for the monkey or fruit for me in order to eat more yogurt. But then some people say, no, I don't. Okay, all right, here. So you bribe the consumer by giving fruit into the yogurt, and then you generate their happiness. All right, but then there are other people that say, no, I don't. I like this gray yogurt. It's all right. I don't care. And so the indifference curves are pretty flat. And then there are other people that say, I really like the gray yogurt. So you're ready to give up some blackcurrant juice to get more yogurt. So you see how these indifference curves play out in the life. Basically, again, the example of how these vectorial choice options are transferred into scalar value. Okay? And then we can do some proper economic tests. We can move on these indifference curves onto different points. And if these indifference curves are actually reflecting the value, that means a higher curve has higher value, then if you know that C, the option C is preferred to the option B in a lower curve, we move around on that curve, and then option C should also be preferred to another option on that lower curve, and that's in fact the case. And then we do a proper transitivity test. When we know that option C is preferred to option B, and so option B is preferred to option A, as this shows here, then option C should also be preferred to option A, and that is in fact the case. So these basic compliance tests work quite well. All right, so we can say that options on larger indifference curves have a higher value than the ones on lower curves, but that the options on the same curve have basically all the same value. So now we can go to neurons. We do our indifference curves here, and then we increase the black current juice, right? This is after the stimulus, after these stimuli, the response of the neuron in the orbitofrontal cortex. We increase the black current juice. That means the animal gives up less black currant juice for getting grape juice. And the response increases and increases and increases just the way it should be. And you see the histograms on the top, they increase from the bottom to the top. They increase with black currant juice amount. Well, there's nothing revolutionary. We know that for hundreds of years. The good reward neurons in orbitofrontal cortex do that. But what we didn't know is this here. So it's not just that the neuron is interested in black currant juice, because we can present an option with only the black currant and compare it to the option that has less black currant and more grape juice. So a neuron that's only interested in black currant juice with lower black currant juice should not have the same response. But we see that neuron does in fact have the same response. So this is a bit of a primitive test, but it shows that the neuron, in this case in orbitofrontal cortex, is in fact integrating the two scalar components of the vector into one scalar signal, right? These different amounts of black currant juice and different amounts of grape juice at choice indifference produce the same neuronal response. And the neuron is, of course, sensitive to these juices, as this example shows. It's not just a stupid neuron that doesn't care. The neuron is very sensitive, right? So we have a value coding in the neuron, and that value reflects both the value from both reward or option components, right? So with that, we can do a more systematic test. Here are our two indifference curves. We place options on this curve. We get this response to this option that contains some grape juice and a little bit of black currant juice. And then we can change the grape juice and the black currant juice, but they are on the same indifference curve, so they should produce, they have the same value, and they in fact produce the same neuronal response. Well, then as a control that the neuron is sensitive at all, we go to the larger option that's preferred to these others because it's on the higher indifference curve, and we see a larger response, right? So this works actually. And now we can really do the experiment. We plot our curves from the behavior. So this is the so-called behavioral indifference curve. Every option on this curve is equally preferred to each other, but preferred to any of the options at the lower curves. And these are the 95% confidence intervals, right? These are the behavioral curves. And then we do the same thing for the neuronal responses, and we plot the same neuronal responses. We fit a function through the neuronal responses of the same magnitude, and we plot these points that we then connect from in this fitted function at the points at which they were tested with the options at which they were tested. And what we see is that these neuronal responses fit exactly the behavioral profile of the scalar values, right? And that is for grape juice. Okay, no, that's all I wanted to say. And then we want to see if how many neurons we need to code this, and whether we can predict the neuronal response and the choices from the neuronal signal. So we have these options on the two indifference curves, and we see that with as little as 10 neurons or so, we can get a 70 to 80% choice accuracy. We have a support vector machine decoder, and we can also get it with the linear discriminant analysis, which also tells us that there's no distinction between responses on the same curve. Okay. Let's see. We need one more. I'm afraid this is going to be messy, but I just never found anything easier. There is a famous axiom from Ken Arrow that says when you change your option set and you remove a non-preferred component in the option set, a non-preferred option from, wow. I'm subscribed to Zoom. We should be able to continue. Can you hear me? Oh my. Can you see my screen? Well, there are two possibilities. The next slides, I only have two more slides. They're not terribly important. And at the end, I have a 3D presentation of this thing, which is very nice. We can, of course, try to log back in this again, but we can also stop here. It's up to you. I have a subscription in Zoom that should. Okay, if you want to. I need to get out of this here. Because I have a Zoom subscription that keeps me running for more than 40 minutes. Yeah. It takes 30 seconds to arrive in my system here. I'm going from wherever you sent it into Proton Mail, and that takes a while. It takes 30 seconds, usually. Are we okay with the time? Oh, here we go. Here we go. I'm unmuted on Zoom. That's correct, I think. Okay. 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 Let's I need to share screen. No, you want to do, it says host disabled participant screen sharing. You first need to authorize me. Yep, I got it. I think we're fine. Let me see. I need to get back into Zoom. I think this is okay. And then I want to go, where were we here? All right, are we back on track? Can you see my screen?
Yes.
Can you hear me? I hear you. Everything is fine. All right. Okay. What I was saying is only a couple more slides. There is this Ken Arrow who says, if you have an option set with several options, you can take away one of the options, that is the one of the non-preferred options, and the preference among the other options should not change. And every economist will tell you that humans regularly violate that principle. And so we thought we'll test it. So formally, the option X out of an option set X, Y, and Z, that means three options, is preferred to option Y in that same option set. If that's the case, then if you remove the option set of that option set, then X should still stay preferred as long as Z is not preferred to X, right? Okay, so we test this. Oops, we have three options. X is preferred, and then we have Y and Z, and they're both not preferred. They're less preferred than X. So we want to remove Z, right? So let's see first. This is the option set with 3, and X is preferred more than 1/3. So X is literally preferred to all the other two options, right? 0.33 would be the average, the chance, okay? Now we remove Z, we have a new option set, the red one, X and Y, and then X should still stay preferred. And that's in fact what the monkeys show. On the left are the behavioral data. 50% would be chance, but X is preferred with 55%. And that's hopefully, I think, is statistically significant, right? So in behavior, the monkeys satisfy Arrow's weak axiom of revealed preference, right? Option X stays preferred. Why do the monkeys do that when humans don't do it? We think that's because they have task experience. They have incorporated all three options into their memory. And when they choose within a reduced option set, they still have kind of that other option set in mind because they have done, let's face it, 40, 50,000 trials, right, over several months. Humans are usually tested with one trial, or if they test stochastic choice with 20 trials or something. Not very much, right? The humans are not used to this and they get fooled. And that's how your supermarkets fool you, or our supermarkets fool us, right? So what about the neurons? Okay, we record the X in the large option set, in the blue option set, and we get a nice response. We record the X in the small option set, and we get basically the same response, right? So this means we are presenting the 3-option random alternation for the blue option set. And when we present look at the X response, then we see a nice response. And then we only have another set of trials, another trial block in which we present options X and Y, but not Z. And the response to the same X, which is physically the same, stays physically the same, right? And that option X stays the preferred option, as the graph on the left shows, right? This is what should be. It follows WARP. Humans do not do that. 60-70% of humans violate that principle. Okay, and now we can do, and this is get messy, control, check the responses to the other options, and they're all pretty similar. That means the less preferred options show a lower response, and there's not much distinction between the different option sets, right? We need this lower response to show that the neuron is actually sensitive to this, okay? So the neural response complies also with Arrow's WARP. And that's the kind of, if you like this kind of conceptual approach, is a very nice result. Okay. There's one more thing. We said that the neuronal value response integrates the two components, or multiple components, I would say, into a scalar signal. But that's not necessarily what one would expect. You could, of course, say, we have a blackcurrant neuron that doesn't care for grape juice, right? It doesn't differentiate between different amounts of grape juice. It just differentiates between different amount of blackcurrant juice. So you get a category of neurons, they're called blackcurrant juice, who don't bother about grape juice. They just respond somehow to grape juice, which is, I mean, it's not quantitative response, so it's not very interesting, okay? And then you could have another type of neuron that is interested in grape juice, but not in blackcurrant juice. Now, these are results. I'm not hypothesizing here, right? So we got two categories of neurons, two populations of older frontal neurons that are specific for particular juices and code their, in this case, amount, probably their subjective value, right? But then you can combine them into a population. And then you can do our plots that we did before of the solid, the neuronal responses will compare that response to the behavioral choice curves, indifference curves. And you see again that with the population of 34, it's probably 34 responses for maybe 25 neurons or so, you get a very nice correspondence between the neurons and the behavior. So we have both possibilities. We have the possibility that the neurons integrate into one single neuron both components, that vectorial to scalar transformation, but we can also have that across the population. Two scalar neurons together produce this vectorial representation. And that is the conclusion I wanted to make. Now we can do MATLAB, of course, and plot these fancy 3D signals, which shows nothing but the increase in grape juice and the increase in blackcurrant juice leads to an increasing neuronal signal. And the signal is similar along this indifference curve, but it increases across the indifference curves. And the conclusion is then the monkeys understand their task, they do meaningful choices, and they distinguish between the rewards. Now, we have other ways to show that, like first order stochastic dominance and second order stochastic dominance and all that. But it's good to know that monkeys can do it. Otherwise, we would be lost when we go to neurons. And then based on these meaningful choices, a meaningful reward processing, we can say that the orbitofrontal cortex has two types of reward signals: the integrated multi-component signal in single neurons doing the vectorial to reflect the vectorial to scalar transformation, and then we can do also a component process in which they do both that across neurons within a population, right? And here are some of the people. This was were my lab members a few years ago. You see Alex, who did actually all this neurophysiology work I was presenting today. Fantastic. We also worked on satiety, which I didn't show. Very interesting change in the indifference curves when the animal consumes reward during the day. And as I said, we were kind of encouraged by Charlie Plott. When I came up with that question, whether we should do it, he was jumping up and down and said, this is really something that you should do. And then we did it also with in humans. All right, I think that's it. Thank you very much. And I'll be happy to answer questions. Hello? Jesus. Hello? Can you hear me? I can hardly hear you. I don't hear you. Hello? Ah, okay. I hear you. Okay, I hear you. Can you hear me? Sure. I should say we're right now running the same experiment on dopamine neurons, and it works in a very similar manner. And they seem to integrate this in the same neurons. Yeah. Are we still connected? Okay.
You hear me?
Very faintly.
You hear me now?
I heard you with a huge echo.
Okay. Okay, you should be able to hear me now. Can you hear me?
Now your voice is back. Yes.
Okay. Um, Thank you so much for the great talk. So in the hall, we have time for one or two questions, but we are going to have a panel for Wolfram upstairs. People who are interested to ask more in-depth questions and get into topics.
Yes. I lost your voice. I didn't say anything. Do you want?
Okay.
Do you want?
So
Can you?
Wolfram, we're gonna have your panel starting in a few minutes. So people are gonna
Okay.
join in into another room. For your talk in that room. Okay.
So okay. Tell me what I should do.
You just stay where you. Yeah, you stay where you are. People are gonna in our side. They're gonna move to a different room, and then we're gonna continue with you. Thank you so much.
Wait. Should we move completely to Zoom, or is it? Do we stay like this? Okay. I'll wait. I'm here. I'm here. No, I'm not going anywhere. Okay. Sure. I'm sorry for all these technical problems.
Um, Hi, can you hear me?
Yes, I hear you.
May I ask a question?
Yes.
Thanks. I had a question about the some uh, some of the slides near to the final slide. My question is that at first we compared the utility with the choice rate, with the rate of preferring a choice.
Yes.
When we were talking about the arrows warp axiom, about that, when we were choosing between two or three options, for example,
Okay.
you showed us a neuron that its response didn't change when choosing between two or between three. My question is that although it's okay with the warp with the arrows warp axiom, but it is inconsistent with the probability magnitude. You know, the choice rate is increased, okay, but it is not. It's increased. Okay, it is over the chance rate. That's consistent with the arrows idea. But why we should still expect that the neural response is exactly to be the same?
What is exactly the question? What is your question?
My question is that first we compared the utility functions with the choice rate, okay? In the last part, talking about the arrows warp, the choice rates are changed, but the neural response is stay the same. How can we discuss this?
Well, the choice. I don't know if you can see my cursor. Can you see the slide?
Yes, yes.
Yeah. So when you see my cursor, the choice is changed because you have two or three options. That means the choice is, of course, always relative, but the amount of the two juices, and therefore the utility of the two juices is identical. It's always the x, right?
Yeah.
Let's give this up here.
So, can you hear us, Wolfram?
Yeah, I hear also the question. See the x. If you, I don't know if you can see my cursor. The
Yes,
x
yes.
is the same, right? And while it is relative to the others, the frequency changes. It's still the preferred option. It's the same. So it would be natural that the response stays the same. The relative
I mean, can you
value,
hear us?
I hear you. I hear the background and I hear a voice.
We
I
can't
hear
hear you.
and you can hear me.
Yes, Professor, I hear you. I
Okay.
asked.
So we can't hear you. I can't. I think you should share the audio too. If it is.
In Zoom or in, I can unmute in Zoom. Now, can you hear me now?
Wait, wait.
Can you hear me now? Okay. All right. I switched on Zoom. Okay. Right. I hear background voices all the time. Okay. Have I answered the question? Hello? Oh, yeah. Well, there was a question, and I hope I have answered the question.
Yes, thanks. Thank you, and thanks, Mr. Alamazad.
It's an interesting question, yes, because the preference is changed, but the choice frequency has changed, but it's also that the option set has changed. Yeah, okay. Right. I have switched on the voice in Zoom, and I can hear you. And if you can hear me, we're probably okay. Yeah. Yeah, I don't know.
Okay, can you hear us now, Wolfram?
Yes, I hear you.
Okay, perfect. So we finally have it now. Okay, so as you can see, the people are sitting in the panel, and they may have some questions. And we have some people online. They may have some questions too. So we will try to manage this. So maybe first people online, if you have questions, you can type
There's
it in.
too much background noise. I cannot understand. There's a lot of background voices. I hear the auditorium or something.
Okay. So first people online, they're gonna ask their questions, and then we're gonna get to people in the room. Okay. So if any person online has a question, please raise your hand. If not, we're gonna have people in the room. People in the room, if you have questions, so you can ask a question and probably can hear you.
Can you hear me now?
I can hear you.
Yes. Thanks for your great talk. I just want to ask one question. I just want to know that if there is any link between reward prediction error and what Professor Friesen said about predictive coding or active inference. They just refer to dopamine as a precision weighting of policy or action selection. And my question is there any link between reward prediction error and predictive processing of Professor Friesen? Or the neurophysiological data of reward prediction error could actually support what they said about dopamine as a precision weighting of policies or not?
Sorry. The link between dopamine prediction error response and what? I didn't understand. What between what and what? Between dopamine prediction error and what?
Precision weighting. So I ask again.
Yes.
Can you hear me now?
I can hear you, but I could not understand the question. And there's always
I said that.
noise.
Is there
All
any link
voices that
between
make
reward prediction
very-
error and the rule of dopamine and the predictive processing theory that or active inference of Professor Friesen that said about dopamine as a precision weighting of policies and action selection? Is these two theories are linked together right now, or there are different theories? I know they are computationally different, but they are conceptualized. I guess they have similarities between them.
I don't know which theory you refer to, but the dopamine signal does not make an action selection. The dopamine signal only tells you which reward is larger and which reward is smaller. Then the striatum may select the action or the frontal cortex according to the value. That means the higher value will then produce the larger the action. The idea of the dopamine neuron is not to make the choice. The idea of the dopamine neurons is to update the neuronal responses in the striatum or the cortex that represent the value, and then these postsynaptic structures in a winner-take-all decision model will make the choice. That means the action selection or whatever, right? The idea, I mean, we can make everything complicated, and you can always get a model working, but the biology says the dopamine signal does not make the choice. The dopamine signal conveys the value, and the postsynaptic neuron take the value and then make the choice that follows the value. Could you hear me?
Yes, we did hear you. Thank you so much.
Okay.
Any other questions? So there's another question in the room.
Professor Schultz, can you hear me?
Yes.
All right. I have two questions. First of all, in almost every reward curve that you show in your presentation, the neuronal signal of the dopaminergic neurons peak at some point, and then almost immediately afterwards, go below baseline. How do you interpret that? Is it normal in every dopaminergic signal to go below baseline after it peaks, or is it in only some cases, and what is it related to?
You mean this response here, the depression?
Yes, something like that.
Well, the background activity is not very strong, right? But when you have an interruption here, it's quite a big change. It's not in terms of percentage. The change, it can never be more than 100%. But it's like when you are in a room and everybody switches the light off, completely off. The room goes from light to dark. That's a big, big change, right? I think that's, if that's the question, that has always been underestimated, I think. This is a 300% positive change. This can never be more than 100%. But that's not the point. The point is to switch off something completely makes a nonlinear difference, a big difference. Hello? Hello?
We can. I think we can hear you, Wolfram.
Okay.
There were some cuts.
Now I can hear you much better. Yes. All right. With echo, yes. With echo. Okay. Go ahead. I did not understand the question. Parkinson's and release dopamine? Oh, yes. I'm sorry. I have a cold. It's cold here. Well, of course, there are a few neurons left in Parkinson's disease, Parkinson's patients. And the best thing is to find a stimulus that produces the largest dopamine response. And that would be a positive reward prediction error, probably. You can also have let's say this one here, the salience signal, right? The salience signal is if you have a loud stimulus like that probably activates many of our dopamine neurons. And there is a thing called paradoxical kinesia in Parkinson patients, in which a patient, for example, can run out of a burning house, although he's totally paralyzed otherwise. That means a very strong stimulus will generate some activity in these neurons. And I would say it's probably this brown-orange response here, the salience response. So I think there are training programs that in Parkinson patients, they stimulate the patient with stimuli. I think TikTok is probably a good way to stimulate the dopamine neurons because TikTok it's a lot of salience, these quickly changing stimuli that produces a lot of salience. Was that? Are you still online? Okay. All right. Okay. Yes, it's for this brown response. The type of stimulus is not important. It needs to create attention. That would be a really attentional. It can be an aversive stimulus. It doesn't matter. Just a big stimulus, something very intense, that would fire dopamine neurons. Okay. Okay.
Okay, I can ask one question, Wolfram. If no one is asking online, I have a question. Okay. So I have two questions regarding the OFC results you were showing. First of all, I want to know how consistent are those equivalency curves that you have, the curves between the currant juice and the grape juice. How consistent are they for the monkey? Does it change from day to day, or are the monkeys very consistent in their preference in a day-to-day experience? Because that's in humans, apparently we change our preference. One day we like grape juice, another day we like currant juice. And for you, it looks like the graphs were very consistent. And second, does OFC have both the single neurons that combine basically their firing follows those curves of equivalence, or do they have more single neurons that are only responding to grape versus black currant? So at the end of the talk, you showed that there are some neurons that respond to one type of reward but not the other, but you didn't tell us what percentage of the neurons are doing what.
Yeah.
Thank you.
The monkeys are very consistent in their choices day by day after a while. Initially, they're not very consistent, and in particular, when the stimuli are more complicated. Normally, these stimuli, the monkey learns these stimuli let me see, I'll show you something. Like these stimuli here with the bars, they learn that in 2 or 3 days. But sometimes it takes a month because people make things too complicated, right? And then it takes much longer. But ultimately, the monkeys learn it and they become very consistent because they do 600 trials or 1,000 trials a day. And then after several weeks, they are very consistent. The neuronal responses follow their consistency. But if then the monkey is sated at the end of the experiment, and say it more on one reward than on the other, then the neural responses change according to the changed value. So they follow the indifference curve changes and the neural responses change. So it's quite amazing, but that takes a while. That the monkey needs to have a good behavior. These are complicated animals. All right. And then at the end, the question of the neurons, I forgot the numbers, but my impression was there were probably more neurons that integrate the two rewards compared to these single specific neurons. But of course, you get a neuron for blackcurrant juice, you get a neuron for grape juice, you get a neuron specific for water, and so on. You get typical neurons. And Camillo Padua-Schioppa has seen that also. And when he changes the selection of juices he shows, then the neural pool also changes. So a blackcurrant neuron is not a neuron that for the rest of the life of the monkey will only signal the blackcurrant. It is just that the neuron signals the blackcurrant at that moment of the experiment in which blackcurrant is being used regularly, right? Okay.
Thank you.
Okay. I have a question. I have two scenarios. I want to test this. So in the first scenario, we have an agent and we have two rewards with different utilities. Reward A has a higher utility than reward B. So in the first case, we first give the agent the reward A, which has the highest reward, the higher dopamine release, and then we give it the reward B. In the second scenario, it's the reverse. We give first the lower reward B, and then we give the higher utility reward A. Now, in which I would expect that in the first case, the amount of reward the amount of dopaminergic signal for the neurons would be lower than the second scenario, because, for example, in the first one, first we give it a higher expectation, and then we are giving it a lower reward. So the neurons
not necessarily, because the different rewards do not predict each other, right? It's not just the sequence, but it must be the prediction that is compared to the reward. Can you still hear me? Because Zoom has switched off now, but we will not change. We can continue. You may not see me. You may not see me. Can you see the screen? Yeah. Let's not start this again. Okay. So what I'm trying to say is, if you have two rewards that follow each other, and they're not predictive of each other, then the dopamine neurons don't worry. If, of course, one reward always predicts the other one, yes, then you have a prediction error, right? It's not the temporal sequence. It's the difference between the reward and the prediction, or between the value of stimuli that have a predictive relationship. Then the signals need to be, the rewards need to be related to each other. Just a temporal sequence is not enough. I'm not sure we have done the experiment, but that's what I would think from all ideas about prediction error.
Okay, you should be able to hear me, right?
Yes, I hear
Correct? You hear me?
Yes, I hear you. It's all fine.
Okay, very good. Very good. Very good. Any other questions from the audience? So, Wolfram, the people who are sitting here are mostly undergrad and graduate students of neuroscience across universities in Iran.
Yeah.
A lot of them are just starting the field. Some of them might be interested in reinforcement learning and some decision making, stuff like that. And as this is a panel, I think any advice from a guru like you, who's been working in neuroscience for close to a half a century maybe or something, 40 years, would be very much appreciated. So if you have any word of wisdom for the kids here, and the ones that might be interested, especially in the type of work you do, the things that they have to do to follow up, that's also would be much appreciated to know about.
Well, not every undergraduate goes into science, right? So I can only talk about science. And I would say science is wonderful because you can do what you like. And although you sometimes have problems getting the experiment working or publishing a paper, which you have all the time, it's a lot of fun, right? So it's not really terrible work. I think the most important thing in science is, of course, you have to understand what you're doing. If you don't understand what you're doing, or you don't want to understand it because you think it's too complicated, then that's difficult. But if you understand what you're doing, then the most important thing is persistence. That means you have to be able to follow the experiment to the end, and then analyze the data to the end, and then publish to the end. If you run away before publication, then you cannot do science. And the other thing is, I would not try to do science only to get publications. Yes, you need publications to have a list, to have a position afterwards. You need a list of publications to have a career, to get positions. But if you just want to make papers, you can do that for 20 years and then you run out of steam. What you need to do is you need to like what you do. You need to pursue it. You need to be persistent, very, very persistent. And then And you will never know what you're doing until you're maybe 40 or 45 years old. You think you know what you're doing, but the big picture, at least I never knew anything. Did this and I did this, I tried this. And only when we had a discussion last night when we went out with the lab, when you know what I was doing, that was about when I was 40 years or 45 years old. It takes a while to get into it. You have to have a lot of patience. What I say, you need to like what you're doing. If you just do it, what's called extrinsic motivation, in order to achieve something like another publication or so, life is very difficult in science. If you like what you do and you can find somewhere a position that pays you a salary and you can keep your family running and everything, and then you can do what you want, and you don't exaggerate your activity, you're not totally busy all day, then you can do some imaginative science, and suddenly you find things that nobody has ever seen before. And that's the fun part of it, right? But it's annoying because you have to get these damn publications. In order to do that, you have to, even if you do an uninteresting experiment, you have to finish it and you have to publish it. And I do that mostly. Many of the experiments you're doing are not interesting. I publish absolutely everything. Every graduate student and every postdoc in my lab had a publication, except one person in the last 5 years, but we're working on that publication also. So I'm insisting, and it's not always easy, in particular for the graduate student and postdoc, to do the publication. They often run away before that because they have no patience. But then I have to do the publications, which I don't like very much because I want to do other things. So persistence and like what you do, I think, are the most interesting thing, most important things. But you got to understand what you're doing. If it's difficult, then I would do something else. All right.
Yeah, that was very good. Thank you so much. Thank you. Those are very useful comments, and I think these are universal problems with the students. They may get discouraged soon because neuroscience is difficult. Doing experiments are difficult.
In the beginning,
but as you said,
in the beginning,
that's right.
Afterwards, neuroscience is not all that complicated, but
that's right.
Okay.
But they have to be patient. Yes. Very, very, very good. Thank you so much.
Okay.
So please use this opportunity to talk with Wolfram. Ask any questions. if you're shy, you can just give us your questions. We're going to translate it to English and ask them. Any topics, anything? So non-scientific questions. Okay, you go ahead.
Well, in science, you want to find new things. You cannot predict new things. So you do work on things that you think are interesting at the moment. If you think you want to work on the magnet reception in the pigeon, then go ahead and do it. Whether it's important or not, that's very difficult to say in the beginning. You can make any discovery in any animal at any time, right? You cannot really plan science in the sense that an administrator or a grant agency plans science. Of course, they will tell you molecular biology and neuroscience is really important. All right, okay. But then what do you get exactly out of an experiment with molecular biology? That depends entirely on you. And you may do 10 years something that's not very interesting. That's the danger. Yes. But of course, in between, while you do that, it's not totally that like in the morning of your 40th birthday, you suddenly know what you're doing. What I'm trying to say is that you gradually realize what you're doing, and then the less interesting thing you drop, and the more interesting things you continue, and so on. That means it takes a while to understand the field. But many people don't have the patience. They want to know precisely what they're doing tomorrow morning. And for that, science can be very frustrating. And those people from my lab so I had about 1/3 of the graduate students that are now in science and 2/3 of the postdoc that are now in science. And when I compare that, those that are no longer in science either could not tolerate the insecurity of what they were doing, or it was just too much work. It can be a lot of work to finish a paper and to 3 times turn around every comma that you're doing until you are happy and the journal and the referees are happy. After 3 or 4 or 5 rejections from different journals with completely idiotic referee comments. I'm sorry, it's very clear. It happens to absolutely everybody, right? So there is a lot of frustration in there. And if you don't think what you do you like, then it's very difficult to justify to continue. And then, of course, you can ask other people what they think about your work, but you'll get a lot of rubbish comments also. Many people told me work on reward is completely wrong, that you should not change the reward, you should study the movement with the same reward, and so on. I said, well, no, it's the opposite. I want to keep the movement the same, and I want to change the reward. And people said, nah, that's completely wrong. You shouldn't do that. Well, you get depressed for 2 or 3 days, you think about it, and then you come back and say, well, it's right, but I still like to do it because of this and this reason, right? You got a little bit kind of gamble that what you're doing is actually useful in terms of creating some novel results or novel ideas. And a lot of people, as I said, will tell you you're wrong. You have to live with that all the time, all the time. As always, an industry that tells you your data are wrong because they are smarter than I am. So they want to show they're smarter, and therefore they tell everything I'm doing is wrong. All right. In the beginning, it was very shocking. Now I'm getting used to it. And after a while, they usually quiet down. I'm sorry, it sounds a bit awful, but sure.
Okay, Wolfram, that was great. That was a great time with you, and it was also, I think, encouraging for students to hear that even you sometimes get rejections from
not sometimes.
idiotic.
Not sometimes. I never had a paper rejected except in the first round in the last year. I don't know before. No, it's the last paper.
I think that's Right. I think it's very encouraging for all the students. So Wolfram is saying he's never had a single paper that was accepted on the first round. He submitted to a journal, and this is Wolfram Schultz.
No, in
So,
the last
guys,
year. I think the last paper that was accepted in the after one in the first journal was, I think, 2017 or something, about skewness with a behavioral paper and PNAS. And after that,
I see. The one that you presented.
I would say 95% of my papers needed at least one journal, two journals, and one paper. In fact, of what I presented today, the behavioral part in humans took 11 journals until it was considered and published. Okay.
I see.
And that was
Very
a
good,
journal by a guy who I knew personally.
I see. I see.
So
I think this is a message of persistence for all of us and perseverance, basically. And as you said, if you know what you're doing is correct, you should keep on doing it and trying to convincing the community that this is a worthwhile work.
And if you're uncomfortable with this, you can think about it a while. But if on the long run you're uncomfortable, I would recommend to do something else. You can do so many nice things with a PhD that are interesting, and it depends on your priorities in life and everything. Yes. But when you do science is fantastic. I would do it again.
That's right. Yes, but you need to have nerves of steel to be able to continue in this field. So I think this was all great advice. It was great to have you virtually. We hope to be in condition in the future to have you in person, Wolfram. I don't know if you visited Iran ever. Have you ever been to Iran?
No, I have not been. I have not.
No. Okay. So I really hope that the political climate soon becomes favorable to inviting people like you, and we can host you in person. If there's anything else you wanted to tell us, we would be happy to hear. Otherwise, the panel is coming to a close.
Yeah. Okay. No, thank you very much for the invitation. It's fun. I told everybody I'm going to give a talk tomorrow morning in Tehran.
Yes.
You want to have fun. You want to have fun.
Yes.
Okay.
Thank you so much, Wolfram.
Well, thanks for the invitation.